Okay, so and this is the capsular space. And this is a body space, so it's lined by parietal and visceral layers. This is the parietal layer on the outside here. And these cells right here are sometimes called visceral capsule, but they have another name called the podocytes, which stands for feet, because they help make these filtration membrane that look like the space between the toes. Okay. Now, and here is proximal convolute tubule. So the fluid leaves from the capsular space to proximal convolute tubule. Then remember these capillaries coalesce into the efferent arteriole and it takes off. Now there's a structure here called a juxtaglomerular apparatus. And you have to know two parts of it. There's a third part that they don't show on the model. And that is there's specialized cells in the distal convolute tubule right here called the macula densa. And they're actually sensory cells for sensing how much sodium there is in the filtrate. But that's going to be a lecture type thing. Here, along here, is are cells called the marginal cells. Some books would call them the juxtaglomerular cells. And together with these cells, plus these cells in the middle called the mesangial cells, which are not shown on the model, helps to regulate the size of the afferent arteriole. And this helps to regulate what's called glomerular filtration rate. That's how rapidly blood is filtered in this structure. That's called autoregulation. But you won't need to know that for the lab quiz. You just need to know these special cells here, the macula densa, and the marginal cells here that are actually specialized smooth muscle cells in the afferent arteriole. And I think that's it for the internal structure of the renal corpuscle. Now, one other thing, I'm going to go show you a slide of kid. Oh, wait a minute, Kayla's still out there. Hang on. What happened to Kayla? Okay, I'm going to share screen here and pull up. Wait a minute. First, I got to go on the web. And Google. Okay. Okay, so if I was going to show you something like up here, you should be able to tell me whether that's cortex or medulla. And the way you tell it is these guys right here, these structures right here. What are these guys? renal corpuscles. Glomerular capillaries in here, capsular space around them. And you only find those in the cortex. So this has to be a slide of cortex. That clear, everybody? And what makes these things in between them, that makes them proximal or distal convoluted tubules. However, can I just point to one and say, oh, that's a distal convoluted tubule or proximal? No, you can't tell. But they have to be one or the other. Versus a different, there's a slide perhaps like this right here. Now this is medulla. You don't see any renal corpuscles, and you see these long, thin tubes in longitudinal section. This is probably a pyramid. It's medulla, and these are probably collecting ducts. So if you see these long, thin, open tubules, and you don't see any renal corpuscles, then you know that you're in medulla. So that's a couple of things that you could, I could ask you if I show you a slide. So that's clear, everybody. Okay. Okay, I'm going to, maybe I'll go get a real kidney and the cat also. May be able to actually see. Well, maybe I could just do that on the web also. Hang on a second. Yeah, this looks like a good one here. All right. It says bringing one, wait a minute here. Can we, that's all right. Instead of bringing one here from the lab, and I'll try to show you on this camera, this is a much better picture of what the dissected sheep kidney looks like. I mean, you go into Google and you put in dissected sheep kidney, and here it is. So you can see this layer, this cortex on the very outside. You can see it's a different coloration than the medulla in here. And you could probably see a pyramid right here. The very bottom of the pyramids are called the renal papilla. And the renal pelvis is this area right here. It's usually filled with this yellow gunk in the preserved kidney. I don't know why. It should be a space, but it's filled with this yellow gunk. And here's the renal capsule right here. It is this thin connective tissue layer on the outside of the kidney, and you really can't see it on the models. They don't put it on the models. What you could also see on the dissected kidney, which is not on this slide, but right here on this one, you can see the ureter. Wait a minute, wait, the list of people is blocking that now. Let me, okay, this right here, ureter. And sometimes also you can see the renal artery and vein, yeah, right here. And this one, you can see renal artery and renal vein. And you can see even from this picture that the renal artery is more circular and more rigid than the renal vein. It's collapsed, it's squashed because it's weaker. It has this smaller tunica media. And there's the ureter right there. So those are the kind of things I could ask. I'll probably ask at least one question on the real kidney. Can I? Hang on a second. What happened to the? There's the list right there. Okay, and then lastly is the cat. Now, what if I can get a dissected cat up on here? Let's see. Yeah. How do I show the, wait a minute here. Or maybe this one's better. So here's a dissected cat. And you can see the kidneys, you can see the renal artery, renal vein, and then this thing being the ureter right there. Going down to the urinary bladder, which is right here. And one thing that might not show is interesting about the kidney. Maybe I'll show it in this one. That the kidneys are not actually in the peritoneal cavity. They're what's called retroperitoneal. They're actually behind the peritoneal cavity. And the covering over the top of an undissected kidney would be actually parietal peritoneum, not visceral peritoneum, which would cover something like the intestines, because the intestines are within the peritoneal cavity. So they have a parietal peritoneum over top, and then underneath that is the renal capsule, because they're actually behind the peritoneal cavity. But the ureter, when it leaves, it eventually enters the peritoneal cavity because the bladder is in the peritoneal cavity, but the kidneys are not. They're, like I said, retroperitoneal. Well, I think that's so I think that's everything you need to know for the urinary anatomy quiz, which I'll be putting up probably like a week from today. Any questions, comments?
I have a question, Dr. Murphy.
Sure.
So the questions you mentioned, there would be at least one on the real kidney. I'm just curious, are most of the questions going to be on cats and sheep parts, or?
No. Most of the questions will be on the models.
Oh, okay.
Yeah. There'll probably be at least one question on the sheep kidney and possibly a question on the cat also. But
Okay.
most of them are going to be on the models.
Okay, thank you.
Yeah. Well, remember to download a version, download group activity 6 from D2L, and that one's on the urinary system. I handed out that in lab this week. And maybe next, and so next week, it's going to be urinalysis. And I guess, and we may be able to work on the group activity, maybe in breakout rooms next week. But, and also, if you got any questions about, remember, group activity 5, some people are asking when it's due. It's not going to be due until next Friday. But if you have some questions about that, or any questions about anything in this time we got right here, just fire away.
Dr. Murphy, do you want to put us in breakout rooms for that, or?
Well, do you want to work on, well, I don't know, if you want to work on a one or more group activities, we can go into breakout rooms, yeah. You guys finished up group activity 5 and 5A. If you have group 6, you could start actually on group 6 if you looked into the urinary system a little bit.
Christine and I had talked about working on 5 a little bit.
Okay, good. Well, let me get a chair. Oh. All right, good. Okay. Sorry about that. I forgot to turn on the recording for the first part of the lab, so I'm going to repeat it now and talk about, this model first. The abdominal pelvic model, we would just have the major parts of the urinary system: 2 kidneys, adrenal glands, renal vein, renal artery, and the ureters going down to the urinary bladder down here. Now, there are 2 urinary bladders, male and female, and they're identical. But they've put some reproductive structures on these bladders which makes the female model much smaller than the male. So if I want to show you some internal stuff, in the bladder, I'm going to use the male model. So I open it up, look inside. And the muscle on the outside is a smooth muscle, but it's called the trusser muscle. The outside is a connective tissue called the serosa, or epithelial. No, it should be epithelial, called the serosa. There's some connective tissue there too. And then on the inside, right here, of the bladder, remember it's lined by transitional epithelium, and it's folded into rugae, an empty bladder, because the epithelium is stratified when it's empty. Now there's a triangular area at the base of the bladder, inside the bladder, that has 3 openings. 2 of the openings are inputs with the ureters. There's a ureter here and a broken off ureter there, and they enter right in here. And then there's one outflow, a tube that's an outflow from the bladder called the urethra. And this triangle made by those 3 openings, that area, is called the trigon or trigone, Trigone. So that's the stuff for the bladder. Then we go to the cross-sectional or longitudinal sectional kidney. The kidney is divided up into the cortex out here and the medulla in here. The medulla has 2 different areas. One called pyramids, which are here, 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 and here, and here. And in between those pyramids are called columns. That's an area in the medulla. Of course, here's the renal artery renal vein, and there are spaces within the kidney, singular called calyx, multiple called calyces, and there are minor calyces. Minor calyces coalesce to form major calyx. The major calyces combine to form the renal pelvis, which is right here, and the inside of the renal pelvis empties into the ureter, which is right here. Okay, then we go to microscopic anatomy and look at the parts of the nephron itself, the functional unit of the kidney. It's a series of tubules, and you need to know what the name of each tubule is, plus the blood vessels that bring blood to these tubules and away from these tubules. Like blood vessels here, called the arcuate artery, arcuate vein, the interlobular artery, and then the afferent arteriole that brings blood into the renal corpuscle, which are right here. These are all renal corpuscles. This is where filtration occurs. These 2 are cut open to see the inside of it. And later on, we look even more closely inside of it in this model right here. Okay, the afferent arteriole brings blood into the corpuscle. It breaks up into capillaries called the glomerular capillaries. Those glomerular capillaries coalesce into an afferent arteriole that goes out and breaks up into a second set of capillaries. This is a porthole system. And strangely enough, the afferent arteriole is called an arteriole, not a portal vein, although I'm not sure why that is. In other portal systems, it's called a portal vein. But anyway, there's a capsule around the glomeruli, and leading from the capsule, this is where blood is filtered. This filtrate leaves the capsule and goes along the proximal convolute tubule in the cortex. Superior to the arcuate vessels is cortex, inferior to arcuate vessels is medulla. And the proximal convoluted tubules go throughout the cortex and start to dip into the medulla on what's called the descending nephron loop or descending loop of Henle. Then it turns right around and goes back up into the cortex along the ascending nephron loop or ascending loop of Henle. This one has a much longer one. It goes way down inside the medulla. Once it gets up inside the cortex, it goes straight to where the afferent arteriole is. And part of the distal convoluted tubule is right next to the afferent arteriole. And then it continues down as a distal convoluted tubule, connects with another tubule here called the collecting duct. And all of these nephrons drain into a particular collecting duct, and it travels back through the medulla way out down here, and finally empties into a minor calyx. Right here, this is the same as this. Once that happens, you actually have urine now. Before that, it's called filtrate. Now remember, the efferent arteriole breaks up into capillaries, some of which are the peritubular capillaries, which are up in the cortex and surround the proximal and distal convoluted tubule. Others go down into the medulla and surround the nephron loops, and those capillaries are called the vasa recta. And they will coalesce into a arcuate vein, which will lead to a renal, and also into a lobular vein, and they'll coalesce into the renal vein and bring blood away from the kidneys. I think that's good on that. Then this, and because I came in, I started remembering to record in the description of this. I didn't even say the afferent arteriole here, efferent arteriole here, glomerular capillaries here, and the capsular spaces here. Remember, this is parietal capsule, this is visceral capsule, also known as podocytes. This is proximal convoluted tubule. This is distal of the same nephron. And remember, these are the macula densa cells here. These are called the marginal cells here. And they're specialized structure, collectively called the juxtaglomerular apparatus, that's going to auto-regulate the size of the afferent arteriole. And this is going to help regulate something called glomerular filtration rate. But that's something we'll need to know in a lecture. You're just going to need to know these structures for the lab quiz. Right? And I think I'm caught up now. I think I even did part of that twice. So I'll put this up on YouTube so you can review it again, and the people who aren't here can actually experience the lab. Okay, I'm going to stop the recording now.